the first session.
Good morning, everyone. It's also a great pleasure for me to chair the first session, and it's very nice to see you all here. Thank you for joining today's meeting, and I would like to thank Andreas especially for the organization of today, which is going to be a very, very busy day. As you see in the program, we will have 5 speakers, and each speakers have 25 minutes. So I would like to ask all the speakers to keep the time, please, and if it is possible, leave 5 minutes for the questions and discussion. And our first speaker is Andreas Thiel. He's from also Charité University, from Medical Department, Division of Rheumatology and Clinical Immunology. Please, Andreas.
So thank you. I have not always been same opinion with Andreas throughout the last 30 years almost, or even longer, 35. So we had some controversies, but I really would like to thank him for setting up this program. Also, not for inviting me personally, but for all the other people, because I really think it's very important to sum up from an immunological perspective, which has not been done always in the last 5 years. So I really thank you, Andreas. I want to start with something, if that switches here. So, historic challenges with novel pathogens. That is my starting slide. This is a very old drawing. So that's what's happening if a really new pathogen meets a population that has never seen this bug. And if no pre-existing immunity exists, this was the example, it is estimated that over 90% have been killed by these Eurasian germs. That is a term I found. So far more than with guns or swords. And there has a term been defined, Native American epidemics. And the reason was that in America, there was limited interaction between the communities, or at least that is thought to be one of the reasons, and limited instances of zoonosis. And in Europe, large number of domesticated animals were in close contact with large human populations, and that led to frequent zoonotic diseases and spreading of these germs. Later then, with worldwide colonization, civilization, highly contagious pathogens also lead to worldwide pandemics. And I give you here a list. You may know this, the Spanish flu, HIV, SARS, all these epidemics, pandemics have happened. And then into 1920, SARS-CoV-2. And what is the common thing among all these different pandemics, endemics? One common characteristic. Without pre-existing memory or cross-reactivity, there were always high fatalities. And without going into detail, I think we all may have read about Spanish flu. Young people preferentially had a high chance to die. And some of the old people were protected by cross-reactivity, existing memory B cells or memory antibodies. So that is, even with vaccines available now, we also should consider that efficient protection for these respiratory diseases has to has to be in the mucosal system. So first contact with SARS-CoV-2. We go back 5 years. And we had some questions whether there's reactivation of pre-existing immunity, because it was known in the beginning that this is a coronavirus and there are some similarities to the common cold coronaviruses. And we ourselves were interested in, so what's happening to the immunity in the elderly? And for most of us, so the first contact looked like that. These are very early analysis, 17 donors, and we looked for T-cell reactivation of all these against all these 4 common cold coronaviruses on 4 follow-up times after primary SARS-CoV-2 infection. And the heat map shows in red, there we could detect reactivated cells against these common cold coronaviruses. So they cross-reacted during this primary SARS-CoV-2 infection. And that happened not to all of us. As you see, there are some donors that only very little, where there's only very little reactivation, but to most of us. And we did something also in the beginning. We just looked for T-cell reactivities against spike in patients, in the first patients here at the Charité, and saw always nice reactivities against the first domain and the second domain of spike. And in most of, or many of the healthy people that were naive with respect to SARS-CoV-2, we saw nothing. But in 1/3, we saw that they reacted against the second part of the spike protein. And to shorten this up, this was due to previous coronavirus challenges with common cold coronaviruses, because in the second domain of the spike protein, there are stretches of strong homology of all coronaviruses. This is the picture of the first, second domain. And in further analysis, we detected that there's a major epitope, at least for CD4 T cells in the second domain. And this is stimulation with this epitope. This is stimulation with the whole S2 part. Almost all of it, at least in the beginning of such a response, was directed against this major epitope. This epitope also serves as a B-cell epitope, has also been published by other people. It's a highly conserved stretch of the S2 domain. And but this is not the only cross-reactivity you can detect, and that is also, we screened the whole SARS-CoV-2 genome or proteome and detected cross-reactivities not only in S1 and S2 or S2 here, also in these proteins, not in these, but here also there's broad cross-reactivity. And the cross-reactivity against these S2 domain correlated with protective antibody titers, just S1 titers, but also neutralizing titers. And that's the data set I really most liked, because it reminded me a little bit of my education in the Institute of Genetics in Cologne a very long time ago, where we learned some basics about primary immune responses and secondary immune responses. And this is the T-cell response against the first domain. It goes up till day 14, and then you reach a plateau. This is healthy donors vaccinated with the mRNA vaccine. And this is the T-cell response against the second domain. And there you see it goes up till day 7, and then already a plateau is reached. And this is what I really like. So these are 30 healthy donors, and they all reacted with a secondary response against the second part of the spike. So very funny. Primary response against the first part and secondary against the second. And what you nicely can see is so these cells dominate the first 10 days, and then thereafter other T cells take over that recognize other parts of the spike S2 domain. So it's not that these immune dominant conserved epitopes occupies all the response against S2 and then nothing else can come up, but rather you can also create then other specificities apparently. I skip this. So a first summary, broad pre-pandemic SARS-CoV-2 cross-reactivity in the population. Responsible, at least when we look at vaccination data, for partly a secondary cellular immune response. And we and others have shown that correlates with stronger and faster humoral immune responses. And also others have looked deeper into that and, for example, found out that in asymptomatic primary SARS-CoV-2 infections, these cross-reactive or other cross-reactive T cells expand. But we still had this open question, so what's happening in the elderly that are highly susceptible in other infections, in other primary vaccinations? We look deeper into that, and that is shown here, which shows that there's decreasing cross-reactivity in the elderly against the S2 domain. S1 is rather nothing happened, and against the control peptide pool also not so much is happening. This is the important thing, that the cross-reactivity goes down in the elderly. So if you're young, you're lucky, you have apparently a lot of cross-reactive T cells, and if you're getting older, really old, the chance is high that you have lost some of them. And the quality, how can we easily access T cell quality with these assays? We invented something we called then later catch, chasing antigen-specific T cells with high avidity, because T cell response quality has a lot to do with functional TCR avidity. What we usually do is we take PBMC or blood, whole blood, we stimulate for a certain time, and then we do something which is very simple. We just look at the CD3 molecule. And that is also, that's old immunology, because it is known if a T cell is strongly activated, the TCR CD3 complex is downregulated. And we just made use of this. Shown here with SEB, a model antigen. But nevertheless, I have to stop this at a certain point. So what you nicely can see, this is gated on CD4 after 16 hours. This is CD40 ligand, so T cell reacting. And this is the CD3 downregulation in these cells. And the funny thing now is, so the T cells that have downregulated CD3 very strongly bear these V beta 19, 27, 28. So the TCRs that are known to be the ones that are really receive this strong SAP activation. And the other V betas apparently are not that activated, not that strong activated. So that also worked with single peptides. And I go through this. And it shows another thing in the old people, the T cell quality response in these cross-reactive T cells goes down. And how does this now end for us? Because this is a long time ago. This is 2020, 2021. Can vaccination or did vaccination reverse this alteration in the elderly? No, it did not, at least not for this special epitope. This is S1 responses after vaccination. This is S2 responses. No difference in between old and young. Young are the blue, the red are the old people. And funnily, we cannot achieve a nice response after vaccination against this epitope. So somehow these cells have gone or are lost or cannot be reactivated by vaccination anymore. There's lots of other specificities also in the old people that can be reactivated against S2, but these are other epitopes. How were the overall vaccination responses, long-term immunity? This is something from the beginning. This is 2 vaccinations, and after 200 days, it doesn't matter whether you are old or whether you are young, titers in the serum go down. But it's no magic, we need a third one. If you do a third one, old and young look similar. You can stay, or the antibodies stay for 200 days, even in the old people, even in the very old people. And but one additional thing is, of course, we not only received vaccinations, but we also received a lot of infections. So hybrid immunity was induced. And that it shows you hybrid immunity as compared 3 vaccinations going down a little bit after 200 days, 3 + 1 infection stays up. Here again, also in the older people. It stays up. And if you compare, now the numbers go down. This is 4 vaccinations going down, or it's looking very heterogeneous. But if you do 4 plus an infection, or if you compare 4 versus 3 + 1 infection, I don't want to argue that infection is better than vaccination, but probably these combinations we now received, most of us, 3 vaccinations at least, and continuous infection does a good job. I skip this. What happens now to these repertoire of cells that cross-react also for other viruses? And this is shown here. This is only vaccination. So number of reported challenges, and here quantity of responding cells. So after 4 challenges with vaccine, you have reacting T cells. But if you compare this to 4 challenges, hybrid vaccination plus infection in any combination you can imagine, then apparently these hybrid challenges reactivate pan-coronavirus reactive T cells. And that also is due in the elderly. This is the elderly after only vaccination, and these are the hybrid challenges. Also, elderly stay a lot higher than if they only received the vaccinations. So next or last summary, hybrid challenges boosted existing SARS-CoV-2 immunity. They preserved SARS-CoV-2 immunity also in the very old people. Very hard to judge now whether these ongoing seasonal or sometimes non-seasonal waves of infections now will substitute for annual vaccinations. Very hard to judge. I think it would need more detailed studies, but I think it's too early to make a really statement. What is one thing at the side is this mucosal immunity, so immunity at the surfaces. The antibodies anyways, it doesn't matter whether infection or vaccination, also after infections, the antibody at the mucosal surfaces don't stay, they don't stay a long time, 2 months maximum, maybe 3 months. But what we show here is that a part of your T cell repertoire apparently cannot be maintained. These cross-reactive mucosal-derived T cell memory. And they can also not be reactivated or this repertoire cannot be re-established by vaccination. Of course not, because it's parenteral vaccination. How can you imagine to re-establish mucosal repertoires? I want to end here So it's still open whether it's now endemic business as usual, because these long COVID ME/CFS risks, for me, it's not really clear. And it's not also clear whether hybrid immunity, so that means additional vaccinations now on top of the infections, maybe exhaust something. So when the summer waves are also not, I think, entirely understood. It would be nothing without my fantastic team, because I was occupied also with other things, with my second child and this research building. Lucy is one of the major scientists, and as I did parental leave, my wife took over and then just did the job further with Lucy. But also all the others here named were very important. And the 2,500 study participants that volunteered repeatedly to give us blood. Without these big studies, I think we would know less. To the BMG for substantial funding. Many other people, also some of you, some of them presenting later, like Eric Zander, for example. And thank you for attention.
Thank you. Thank you so much for the great talk. It's a huge population. Actually, I would like to ask one question first. How do the functional characteristic, for example, cytokine secretion or proliferation capacity of cross-reactive T cells compared to SARS-CoV-2 specific T cells generated during infection or vaccination? Have you checked?
At least the functions we checked, which was mainly cytokine secretion, did not differ that much in the situations where we were able to compare truly only vaccination-derived cells with cells initially primed in the mucosa for any reasons floating around, and then getting the vaccination reactivation. Not so much differences. Therefore, I think the design of our studies was not helpful. And now it's over. Everyone has a mixture.
Okay. Is it possible to follow up these patients?
Again,
is it possible to follow up these?
Yeah, we have still.
Do you still carry on?
We still carry on without money. As all the other lab leaders here know, sometimes even without money, you go on.
Okay. Any question or comments, please, Ursula? Sorry, the microphone is coming.
Microphone is better, I think.
What was the question? Who was asking the question? You?
Thank you very much for this very nice presentation. I'm just wondering what is now your resume on the protective value of the T cell responses. I mean, this is a sign that the immune system had contact, but it doesn't necessarily tell us about the correlate of protection. This is one question. And the other one is, do you think that in elderly, it was due to the mRNA vaccine that this was a reason why not the pan-corona epitopes could be activated, but probably another type of vaccine, like a whole cell vaccine in combination, would have given the chance to reactivate this pan-corona epitopes in elderly as well before they got infected.
Now, second one first. No, I don't think that any other parenteral vaccination can really trigger the mucosal cells or make any difference to mRNA. We worked before on elderly versus young people and flu and yellow fever vaccinations, no one of us expected these fantastic responses in people 90-year-old. This was a totally surprise for me that a vaccination after 3 shots, you have long-term T cells even in 90-year-old people. So from this point, but the first question again, sorry.
The correlative protection value of the T cell responses.
No, I think there's no definite proof, but the fact that you have T cells that can respond within 7 days and help B cells, I think this is, for me, so logical. This makes a difference. And we have a little data on that. We don't have 1,000 people. That would be nicer to show that, yeah, the antibodies are faster and that kind of things. But everything, I think, points into that direction. Yes, this is good. It is really on days. It's a competition, virus or antibodies in the end. Carsten.
Okay.
Yeah, very nice. So I was just wondering, I mean, you showed that I think in the older or in the elderly population, the mean of the antigen-specific T cells was the same, like after the 3rd vaccination, but the spread was enormous. So you have some people who have really high responses, and so that obviously some people have a problem. And what is the correlate? What is the predicting basically whether you have a good response or not a good response?
I think it's very hard to look at T cell data and say, okay, if you have 1 in 10,000, this is worse or this is bad. And if you have 10 in 10,000, this is good. From yellow fever, we know that 50-fold, 100-fold differences in the count in the blood don't make a difference. In the end, all these people have been tested for antibodies, and they're all, at least after 3 vaccinations, beyond really high antibody induction. So the amount of T cells you see is not really reflecting good or bad response, but what these T cells can do in the end. And even though in the old people it is heterogeneous, maybe in 1,000 people we have 10 donors that don't respond very well with antibodies.
Andreas, I wanted to come back to this pre-existing immunity. So in SARS-CoV-2, you nicely showed that in the elderly, this was gone already, and you couldn't or it was lower, and you couldn't really reactivate it with a vaccination again. So I guess the interpretation would be that those T cells are gone because of overstimulation or whatever. But then coming back to the Spanish flu, where the elderly did have pre-existing immunity. So why was that pre-existing immunity not gone? So what's different? Is there something different between the flu and SARS-CoV-2, or are there other factors that we don't have taken into consideration yet?
A good point. I cannot answer. I can just say that these few cross-reacting T cells we still observe in the elderly, they are not exhausted. We did single cell analysis. They look like it's just gone. But I cannot argue, okay, what is then the difference? Yeah.
Okay, please, from the back.
Can you hear me? What would be your conclusion with respect to vaccinee age of vaccinations that have been at the moment just recommended for the elderly? Would you because you have basically 2 messages, so the high affinity T cells are more or less gone, but the more often you vaccinate, the higher the likelihood that you reach some plateau also in the elderly. Would you recommend that vaccinations against pneumonia or herpes viruses that are recommended for the 65-year-olds would need to be administered earlier?
The first point, high affinity cells are gone, is not true. Only the cells recognizing this one epitope there's something different. The vaccination is able to induce a lot of other T cells against a lot of other epitopes, apparently, also in the very old people. I think we don't know everything, what is now happening after mRNA, and how long does this stay. The problem in all these vaccinations is mucosal antibodies, which are induced also after mRNA vaccination, don't stay a long time, 1 or 2 months, and then they are gone. And then you have this infection challenges on top. But even after infection, and many people know that, you can be reinfected after half a year. So it's this reinfection, if you get a high dose of virus, yeah, and your antibodies at the mucosal surface are low, yeah, you will get another infection. But you will respond fast because you have T cells, you have memory B cells, they adapt because they mutate again against, yeah.
Okay, last question, please, Christine.
Andreas, was really nice and deep analysis into the age component of the T cell responses. Everyone saw that in the severe cases, these were associated with severe lymphopenia and T cells disappearing to 90% loss in the periphery. And it was not clear how that really did recover. Did you have cases where you had lymphopenia and you saw recovery because the percentages may be stable, but the total numbers might differ quite substantially? And if so, would you think that we sort of lost kind of the repertoire due to this very strong specific responses against SARS-CoV-2, and then other sort of naive T cells were sort of at the costs of naive T cell responses and the repertoire?
Didn't look deeper into that. Can just refer to the to other publications that looked in the innate system, that if the early interferon alpha response isn't that good, then something is happening with innate cells like myeloid cells, and they behave very strangely. And then, yeah.
Related with these, any of your patients had primary immune deficiency? Have you checked the other clinical?
It's mostly healthy donors. And
yeah.
Of course, if we would have seen something like strange lymphocyte subset counts, we would have looked at that deeper, but
okay.
not the case.
Okay, thank you so much.
Thank you.
Thank you.